Thank you so much for the invitation and for the really kind introduction. I'm delighted to be able to contribute to the seminar series. And as you mentioned, I'll be talking about how we can use cardiovascular imaging in population science. And I'd like to start with a bit of background on cardiovascular epidemiology. Now, epidemiology is concerned with describing the distribution, frequency, and determinants of disease. The very first large-scale study of cardiovascular epidemiology was the Framingham Heart Study. This is a population study which recruited the residents of a small town in the US called Framingham in the USA. And this study resulted in some really seminal findings. It was the first study that highlighted major cardiovascular risk factors such as cigarette smoking, high cholesterol, high blood pressure, and lack of exercise. The study itself was launched in 1948, and since then, subsequent generations of the Framingham cohort have continued to contribute to the project. Since this start to cardiovascular epidemiology, we've seen the emergence of large biobanks and national efforts to organise and link up routine health data across many countries in the world. In the UK, Health Data Research UK has a number of really ambitious programmes to link up population level data in the UK and present these in a way that it may be useful for biomedical research. There are also dedicated research cohorts such as the UK Biobank in the UK and elsewhere in the world, such as the Kadoorie Biobank in China, the Taiwan Biobank in Taiwan, and the MESA cohort in the USA that present a detailed study of individual cohorts. And many of these research cohorts, as well as having detailed information about environmental exposures and clinical outcomes, are starting to incorporate large-scale cardiovascular imaging. For example, the UK Biobank, the SHIP study in Germany, and MESA all have large-scale cardiovascular MRI data as part of the resource. And this information to this scale has never been available to us before, and so presents unique and new opportunities for cardiovascular research. Now, let's think about why the imaging data might be useful. We can use the cardiac imaging data to derive measures of size, structure, and function for each cardiac chamber. And this can give us an indication of health or disease at an organ level. And we're able to do that across the entire sample for each of these cohorts. And this can be something that's really powerful. It can give us a more complete picture of cardiovascular health compared to if we're just thinking about clinical outcomes, and gives us a much deeper understanding of the health status of our individual. Many of the imaging indicators also become apparent before the onset of clinical disease, and so they can be really useful as early or preclinical indicators of disease. Now, I'm going to talk to you about my experience, and this is mostly in the context of UK Biobank. So I'll start with a bit of background to UK Biobank, and then I'll take you through some things that I want to highlight to you within that. So the UK Biobank, as many of you will know, is a very large UK-based cohort study. It recruited just over 500,000 individuals from across the UK over a 4-year period between 2006 to 2010. All of the participants underwent a very detailed baseline assessment, including characterization of their demographics, lifestyle, health status, a series of physical measures, and blood sampling. In 2015, the UK Biobank Imaging Study was launched, and this includes multi-organ imaging for 100,000 of the original participants. The imaging protocol includes imaging of the heart, brain, abdomen, carotid ultrasound, and whole body bone densitometry. In 2022, the UK Biobank Repeat Imaging was launched, and the idea here is that a subset of the individuals who completed the first imaging visit will have repeat imaging. Importantly, the UK Biobank has established extensive health record linkage for all of its participants, and this includes linkages to hospital episode statistics, death registration data, cancer registration, and primary care data. And this information is periodically updated by the UK Biobank and allows prospective tracking of incident health events for the entire cohort. And this information about health outcomes can be linked back to everything that was collected at the baseline visit, as well as all the information that's been collected at the imaging visits. This is what the imaging data looks like from a cardiac perspective. So this is a sample of the cardiac MRI data. And here you can see a short axis slice of the left and the right ventricles. You can appreciate the contraction of the left and right ventricles. You can see the chamber volumes and also the thickness of the walls. For this information to be useful for us in an analytical sense, we need to turn the imaging into numbers. So we need to derive phenotypes that we can quantify and use in subsequent analysis. If you were doing this in a clinical setting, you would sit down with a post-processing software and you would do some measurements, and this involves some manual interactions with the scans. But when we're thinking about a resource like UK Biobank, where we are faced with the task of analysing thousands of images, that kind of manual approach is simply not feasible. And there is need to develop automated methods to handle this level of data. And for this reason, there has been development of automated analysis pipelines to allow derivation of imaging phenotypes. There are dedicated pipelines that have been developed and validated in the UK Biobank and are now available for use. And in this way, we can derive our imaging phenotypes in an automated and scalable manner for the entire imaging set. And this is a sort of example of measurements that we can derive. So, for example, we can obtain measurements of volume of all 4 cardiac chambers. We can have measurements of the myocardial mass, so how much the amount of mass in the left ventricle. We can also obtain more advanced measures of shape, such as a concentricity index. We have measures of chamber function and also measures of and myocardial character, such as native T1, which I'll talk to you more about later in the talk. This imaging data can be really helpful in allowing us to build a picture of organ level alterations associated with various health exposures. We can also use the imaging information as biomarkers for disease detection or prediction, and we can also use this information to better understand underlying disease mechanisms. And what I'd like to do in the rest of the talk is to share with you 3 exemplar studies that illustrate each of these 3 points. So firstly, how can we use the imaging data to build a picture of organ level alterations? This is a recent study that we did in our group, which was focused on better understanding the cardiovascular phenotype of individuals who have a diagnosis of hypertension. For this study, we used just over 39,000 participants from the UK Biobank who had CMR data available. We ascertained the clinical diagnosis of hypertension through looking at all the health record databases that were available. And we examined the associations between hypertension versus no hypertension with a range of selected CMR metrics using multivariable linear regression adjusting for major vascular risk factors. And this is what we found. We found that individuals who had a diagnosis of hypertension compared to no hypertension had a larger, poorer functioning left atria, that's the top chamber of the heart, a thicker, poorer functioning left ventricle, that's the main pumping chamber of the heart, and they also had lower aortic distensibility, which indicates greater stiffness in the main arteries of the heart. These are all measures of adverse cardiovascular changes, and this level of detail of how hypertension may impact different structures of the heart had never been described before in the context of hypertension. The next thing we were interested in was whether the associations of hypertension with cardiovascular changes varied across different subpopulations of our sample. And so we looked at differences by sex. What we found was that the relationships between hypertension and cardiac remodeling were roughly similar in men and women. However, we found that women had a greater decline in aortic compliance, so more stiffness in their big arteries in response to hypertension than men, suggesting that they are perhaps more severely affected by exposure to hypertension. We then looked at potential differences across ethnic group, and here we found that individuals from Black ethnic groups had a greater increase in left ventricular mass in association with hypertension than any other ethnic grouping in our sample. Then we were interested to understand the influence of having good blood pressure control compared to poor control in those who had a clinical diagnosis of hypertension. To do this, we referred to the physical measurements of blood pressure that were recorded as part of the UK Biobank assessments. And we classified our individuals who had a diagnosis of hypertension into good control versus poor control based on those readings, and comparing those to what their blood pressure should be according to national guidelines. And then we simply ran our models again in those subgroups. So clinical hypertension with good control of the blood pressure, and here we have clinical hypertension with poor control of the blood pressure. And the differences are really quite striking. If you look at the panel on the left-hand side, these are individuals with hypertension and good blood pressure control. We find that many of the associations between hypertension and adverse cardiovascular remodeling are attenuated. And this presents really strong evidence of benefit from optimizing blood pressure control in individuals with hypertension. And so, to summarize, this is the largest imaging study describing cardiovascular alterations in association with hypertension, describing how hypertension affects different structures within the cardiovascular system. We demonstrate differential relationships across patient groups, which may be used to better understand differential risk in different patient populations. And also, we clearly demonstrate the benefits of good blood pressure control in our sample. Now, the second study is focused on demonstrating the value of imaging phenotypes as potential biomarkers of risk. And for this, I want to focus on myocardial native T1. Myocardial native T1 is a special MRI technique which allows us to calculate the longitudinal relaxation time of a certain tissue and display this value as within a voxel in a parametric map. The T1 value is influenced by the magnetic property of the underlying tissue. And so the number that you get for T1 can reflect what the underlying tissue is made of, and so its underlying composition. And so it can give you an indication as to whether there is fibrosis of the underlying heart muscle, infiltration, or scar. And as you can imagine, this information can be really useful about understanding, firstly, differences between health and disease, but also even distinguishing between specific disease entities. And some people have talked about T1 as being almost like a non-invasive biopsy of the heart, so potentially a really powerful imaging technique. Up until now, native T1 has been examined within selected clinical cohorts that have specific cardiac conditions. For example, individuals who have specific cardiomyopathies such as HCM or hypertrophic cardiomyopathy. And its role within a population setting is not well understood. This is information that is acquired as part of the UK Biobank cardiac MRI imaging protocol. And this group in the University of Oxford, led by Professor Piechnik, have developed an automated method for extracting native T1 values for the UK Biobank imaging set. And we were really lucky in that we had the opportunity to work with them and to work with the data that they had generated. I'm not going to go through the entire paper here, but I'd just like to share with you the main findings. These are our results looking at the relationship between native T1 and incident cardiovascular diseases and mortality outcomes. In this figure, you can see results from Cox regression models, from models that have native T1 set as the exposure of interest, and the outcomes listed on the left-hand side set as the model outcomes. The left-hand side panel is a minimally adjusted model, including adjustment for age and sex only. And on the right-hand side, you can see results for a full confounder adjusted model. and the bars are hazard ratios with one standard deviation increase in T1. And so we can see that higher levels of myocardial native T1 were associated with a higher incidence of a range of cardiovascular diseases and with mortality outcomes. And importantly, we found these relationships to be unchanged, even when we limited with individuals who had no evidence of clinical cardiovascular disease at time of imaging. And so in this study, we showed for the first time that native T1 would be could be a useful predictor of future cardiovascular events, even if in people who did not have disease at that time. And thirdly, I want to share with you a story around using imaging as a method to better understand disease mechanisms. And for this, I want to talk to you about pericardial adiposity. This is something that we became very interested in because of really interesting mechanistic and biologic work that was being generated in this area. Pericardial adiposity or pericardial fat refers to these deposits of fatty pockets around the heart that are sort of hugging the heart. You can see them in these panels here on the left. A number of animal studies and biologic studies have demonstrated that there is something distinct about this fat. It has distinct pro-inflammatory properties and metabolic properties that drive the occurrence of cardiovascular diseases such as ischemic heart disease and heart failure. Most of these studies have been focused either on small animal samples, and those in humans have been focused on small cohorts with either particular conditions or small cohorts of healthy individuals. And we started to think about whether pericardial fat was something that we perhaps could measure on the imaging data that we had available to us. Up until this time, there was no standardized way to measure pericardial fat using imaging, and there was certainly no automated way to do so. And so we decided to create this. And after examining our images, we experimented with lots of different ways to measure pericardial fat. And we came up with a standard operating procedure that we felt worked best according to our initial experiments. So this is the method that we used in the end. The figure you can see here is a long axis cut from a cardiac MRI measure, MRI scan, showing the left ventricle here and the right ventricle here. And this white area around the ventricles is an area of fat. You can see it on this picture labeled A, and then in the bottom picture, this is a scan of a different participant. And again, you can see this kind of halo of fat around the heart. And the method we went with was really simple. We just simply drew around the area of fat, and this allowed generation of an area measure. But of course, we needed to automate this process. And after creating a ground truth manual segmentation data set, we used this to develop an automated tool using a convolutional neural network, which generated these area segmentations, outputted the area metric, and also had a quality control indicator, which allowed us to understand the quality of the segmentation produced. This tool was developed in the UK Biobank, validated in an independent holdout set within the UK Biobank, and also within an external independent set. This work is published in a separate technical paper, but it allowed us to generate pericardial fat measures for the entire UK Biobank imaging set. And we ended up with images that looked like this. So we had the segmentations, the area measure was outputted, and then we had this DICE score, which was our indicator of segmentation quality. A DICE score of 1 would indicate perfect segmentation quality, and anything lower than 0.7 we considered poor quality and didn't include in subsequent analysis. This information allowed us to characterize for the first time the distribution of pericardial adiposity in a population cohort and to describe differences in pericardial adiposity deposition across sexes and different age groups. And what we were really interested in was to understand whether the amount of pericardial fat was related to the likelihood of important cardiometabolic diseases, and whether these relationships were independent of other measures of obesity. And these are the main results from that analysis. So here we are seeing logistic regression models with pericardial adipose tissue area set as the exposure of interest. The outcomes are the conditions that are listed on the left-hand side, and we have two different levels of adjustment. The covariates are listed here. The green line represents the fully adjusted model, which includes all vascular risk factors, demographics, and detailed measures of physical measures of obesity and also other measures of fat from body composition methods that I'm not going to go into. But essentially what we find is that higher levels of pericardial adipose tissue are linked to higher odds of having diabetes, hypertension, and high cholesterol, and that these relationships appear to be independent of all the covariates that I've mentioned. We also looked at the relationship between pericardial adipose tissue and blood biomarkers, and here we found that greater pericardial adiposity was linked to an adverse serum lipid profile, poorer glycemic control, and higher levels of systemic markers of systemic inflammation, such as higher levels of C-reactive protein and lymphocyte count, and also higher levels of pericardial fat was linked to higher levels of markers that have previously been linked to cardiovascular risk, such as cystatin C and urate. Furthermore, we found that higher levels of pericardial fat was linked to an adverse remodeling of the left ventricle, the left atrium, and the systemic arteries, and that this was independent again of other measures of obesity. We found that higher pericardial fat was linked to a thicker, poorer functioning left ventricle, smaller, poorer functioning left atrium, and stiffer central arteries. So this study allowed us to demonstrate that higher pericardial fat was likely linked to higher risk of cardiometabolic and cardiovascular risk in a way that was independent to other measures of obesity. We then reflected on the biologic studies that I mentioned at the beginning of this story, and in particular, the ideas that it was the metabolic activity of the fat that was important in driving its relationship to cardiovascular risk. And we wondered whether we could perhaps capture that metabolic activity or character of fat using our imaging techniques. And to do that, we designed a study where we extracted even more information from the imaging using a post-processing technique called radiomics analysis. Radiomics is an analysis technique which allows extraction of highly detailed information about shape and character from a defined region of interest. So for us, the region of interest on our image was the pericardial adipose tissue area, and from that, measures of advanced measures of shape were extracted, and the signal intensities within this region were used to derive radiomics measures of tissue character, essentially. We then used these extracted radiomics features to develop models for prediction of clinical incident heart failure. This analysis demonstrates that models using pericardial adipose tissue radiomics showed good performance for prediction of incident heart failure. And then we examined which features within the model were important in driving our outcome. And in doing so, we demonstrated that it was both the amount of fat as well as its character that seemed to be important in relating to heart failure risk. And this corresponds really nicely to our biological understanding of the relationship between pericardial adiposity and cardiovascular risk. And so our results here are so far are very suggestive that there is a distinct mechanistic role for pericardial fat in driving cardiovascular risk. And the next thing we wanted to examine was whether there were distinct genetic markers that were determining the proportion of pericardial fat deposition. And in the UK Biobank, there is also genetic data available. And so we performed a GWAS with our newly extracted pericardial adipose tissue phenotype. And in doing so, we identified multiple distinct genetic loci associated with pericardial fat area. And this was after accounting for multiple measures of whole body adiposity. Our results showed that the proportional deposition of pericardial adipose tissue is to an extent genetically determined, with a hereditary component estimated at around 9%. This was then a really good opportunity to examine the causality of the observational relationships that we had previously demonstrated. And so we used the variants that we had identified as genetic instruments in Mendelian randomization analysis, where we identified likely causal relevance of genetically predicted pericardial fat with adverse CMR measures of left ventricular structure and function consistent with our previously described observational relationships. So those were the three studies that I wanted to share with you as exemplars to illustrate the potential of imaging in enhancing the way we study cardiovascular disease across different themes. So to conclude, the availability of research resources with large-scale imaging banks presents new opportunities for cardiovascular research. Integrating image-based information can improve our understanding of the impact of exposures on organ-level health and allow us to delineate disease mechanisms. Imaging biomarkers may have a role in improving risk stratification and targeting of preventive therapies. Thank you so much for the opportunity, and I'm happy to discuss and take any questions.